There is a concept, or principle rather, that can be applied within any discipline, but it is especially applicable within the natural sciences. It is known as the precautionary principle. It is one that may seem confusing or unclear at first, but once you have a few examples, it will seem like common sense. The precautionary principle states that we should not take action on something unless we can verify the potential benefits outweigh the potential harms. Let's look at an example of the precautionary principle in context. Climate change is a widely accepted phenomenon by the scientific community, even if certain world leaders and their appointed officials, members of elected office, and other public figures openly deny it. There is consensus that there must be action on climate change, and the actions would help reduce greenhouse gas emissions. In turn, vulnerable habitats in polar regions and islands will be conserved, storms and natural disasters won't keep getting stronger, and it provides jobs by creating the need for innovative solutions. However, action will also mean the loss of jobs, delay economic growth for developing nations due to limited access to resources and new technologies, and challenges in enforcing action internationally. Oftentimes, the rules of science and experimentation come as an afterthought, but you can use the precautionary principle in designing your experiments to limit the stress and harm on test subjects. The International Baccalaureate Organization has published its official statement in 2015 about the use of animals in experimentation. They can be remembered as the 3 R's: replacement of animals with cells, plants, or simulations, refinement of methods to reduce distress, and the reduction of numbers. Experiments involving animals must be based on observing and measuring aspects of natural animal behavior. We must follow these guidelines due to the ethical considerations with animal testing. In terms of rules with human experimentation, the Nuremberg Code was established in 1947, setting the following ethical requirements. Informed consent for experiments. Experiments must be scientifically necessary and conducted by qualified personnel. Human trials should be preceded by animal studies and surveys of a disease's natural history, and benefit to science must be weighed against risks and suffering of experimental subjects. You might be thinking, that's too many rules and things to consider all at once. What's the need to experiment anyway? It is the way knowledge is created in the natural sciences. We have ideas and questions about the matter and energy that surrounds us, and the only way to establish those ideas and answers as facts or accepted truth is to test them. Sometimes knowledge is gained by accident, like with penicillin. Penicillin is an antibiotic substance synthesized by the mold Penicillium. Sir Alexander Fleming was working as a bacteriologist at St. Mary's Hospital in London. In 1928, he happened to be working with Staphylococcus aureus being cultured in petri dishes. Staphylococcus aureus is a bacteria naturally found on our skin and can cause minor skin infections to life-threatening conditions. After returning from his summer vacation in September 1928, he recognized the mess that he left. One culture of S. aureus happened to be uncovered and was contaminated with a mold. Upon closer inspection, Fleming observed a zone around the mold that inhibited bacterial growth. He soon identified the mold as Penicillium. He cultured and experimented with it some more, and then he was able to conclude Penicillium produces some factor that inhibits bacterial growth and reproduction. On February 14, 1929, Fleming introduced penicillin to the world to cure bacterial infections. As Dr. Fleming famously wrote about that red letter date, "When I woke up just after dawn on September 28, 1928, I didn't plan to revolutionize all medicine by discovering the world's first antibiotic or bacteria killer. But I guess that was exactly what I did." It would have to be tested, though, to conclude whether penicillin was safe for human consumption in curing infectious disease. When knowledge is created in the natural sciences, it is up to scientists to continue experimenting with it to develop as complete a model as possible. Scientific theories and models are only valid as long as they can explain all of the available data. This is why we must collect sufficient data through which we can then evaluate the hypothesis. In the final part of science lab reports, students write about the strengths and limitations about the research and experimentation. Oftentimes, students write about the strengths and limitations related to the fidelity of how the procedure was carried out. But what should certainly be addressed through these is how well the experimentation modeled the theory being tested. This helps us with refining and changing models so they more accurately reflect the relationship between parts in a system. Models are the building blocks of science. In fact, it is the building block of science and the scientific method. We use the scientific method as a model to compartmentalize the steps and understand what we are supposed to do as we are moving from asking questions to posing hypotheses, designing an investigations procedure and apparatus, collecting data, processing it, interpreting the results, and evaluating all aspects to communicate the results and refine and/or revise the investigation for further research. Basically, a model is a substitution or a simplification of a system or a stand-in for a prediction. The key part to a model is it has to be similar to what it represents. An experiment is a model, and it can only represent a real phenomenon to a certain extent. This is why replicates in experimentation are so important, because it helps us determine the strengths and limitations of the model and identify how the experiment should be modified to show more of the relationships between parts in the system. The more times you test a relationship and the more you collect consistent results, the stronger your model will become. Going back to the discovery of penicillin, Fleming posited that penicillin would work nicely as a bacterial inhibitor. This still needed to be tested before it could become available to the masses, starting in 1942. In March 1940, Australian pharmacologist and pathologist Sir Howard Florey and British-German biochemist Sir Ernst Chain began clinical tests to purify and distribute penicillin to treat and cure many infectious diseases. Florey and Chain did a series of clinical tests at Oxford University to conclude the effectiveness and safeness of penicillin for treating diseases. The first test was done by injecting a highly concentrated dosage into 2 mice. Despite the high dose, the 2 mice appeared to be unharmed. Then, to test the effectiveness for the use in treating infectious disease, they decided to increase the amount of mice tested from 2 to 8. All 8 mice were deliberately infected by hemolytic streptococci bacteria. 4 of these mice were then injected with penicillin, and the other 4 were not. 16 hours later, the 4 injected with penicillin survived, and the other 4 died. In early 1941, they felt ready to begin testing on humans. The first patient, Albert Alexander, a 43-year-old policeman, was treated with penicillin on the 12th of February 1941. Albert had a scratch that developed into a pretty terrible infection. Within 24 hours of being given penicillin, his health improved. However, the stock of penicillin ran out before the full course of treatment was complete, so Florey extracted excess penicillin in Albert's urine and re-injected it. A month later, Albert's infection relapsed, and he died in April of 1941. As Florey concluded about this, treating and curing infections in mice was one thing, but humans are roughly 3,000 times bigger and would need 3,000 times more penicillin. As a result, their laboratory was turned into a penicillin factory, production running 24 hours a day. These 2 men continued their trials with a few more human subjects. One was with a 4-year-old boy with an eye infection. He was cured of the infection but later died of brain hemorrhaging caused by a weakened artery from that infection. When these trials demonstrated that penicillin was effective in humans, they pushed for the mass production of the antibiotic. In 1942, penicillin was mass-produced and available for public consumption as treatment for bacterial infections. If Florey and Chain were alive today, their experimental method would not have passed approval. Risks involved with human testing are minimized with strict protocols. These protocols require multiple tests on animals over a long period of time. After it passes checkpoints, it can then be tested on a few target patients. Following deep analysis of results and assessment of the risks, it can then pass to trials involving large numbers of patients to check there are no severe or common side effects. In evaluating their drug tests, Florey and Chain tested penicillin on mice before testing on humans. While the results were very clear, they really had limited testing on mice before risking use in humans. However, the first humans that were given penicillin were close to death and did not investigate potential risks with healthier individuals. The initial samples of penicillin were very impure, particularly with Albert Alexander's treatment. Additionally, there could have been toxic substances in the samples. In the end, though, Sir Fleming, Sir Florey, and Sir Chain were awarded the Nobel Prize in Physiology or Medicine in 1945 for the discovery of penicillin and its uses in treating infectious diseases. While the protocol was flawed, they did get lucky with the successes in penicillin research in developing the world's first antibiotic.